Immunohistochemistry and fluorescence microscopy are incredibly powerful tools that neuroscientists use to identify neurons according to specific traits. Immunohistochemistry is the process of using antibodies targeting specific proteins or antigens expressed in a particular cell or particular compartment of the cell. Typically, a primary antibody is applied which recognizes the protein of interest. This is followed by a secondary antibody, which binds to the primary antibody. The secondary antibody can be tagged with a molecule that emits fluorescence under specific conditions. Using a fluorescence microscope, you can then see which cells in a sample of brain tissue emit this signal, indicating the presence of the protein of interest. For example, in this sample, immunohistochemistry and fluorescence microscopy have been used to identify dopamine neurons in the substantia nigra. There are many ways to use these tools to better understand the structure and function of the brain. So let's take a look at the specific steps involved. There are 4 basic steps in immunohistochemistry and fluorescence microscopy. First, the brain is cut into very thin sections. Second, those sections are cycled through a series of washes followed by incubations in a solution containing the primary antibody, an antibody that binds a protein of interest. The sections are then incubated in a solution containing the secondary antibody, an antibody that binds to the primary antibody. This secondary antibody is tagged with a fluorophore, a molecule capable of emitting a fluorescent signal under specific conditions. Following these incubations, the labeled tissue is mounted onto microscope slides. Finally, those slides can be examined with a fluorescence microscope. A fluorescence microscope transmits a light of a specific wavelength through the tissue. This light is capable of exciting the fluorophore, which then emits a different wavelength of light. Using a series of lenses and filters, researchers can use this microscope to visualize all the cells in the sample that express the original antigen or protein of interest. Here you can see a whole mouse brain that is being trimmed in preparation for mounting in a vibrotome. As the name implies, the vibrotome slowly moves a vibrating blade through the tissue, cutting very thin sections, typically between 10 and 100 microns in thickness. Once the brain has been sliced into sections, those sections can be collected into wells containing buffer solutions. Once the tissue sections have been collected, they are cycled through a series of washes and antibodies. Here, we are using wells with mesh bottoms to easily move the sections through the relevant washes and antibody solutions. The first antibody to be applied is called a primary antibody. In this particular experiment, we want to identify neurons that are expressing CFOS, a protein product of an immediate early gene that is produced when the cell is highly active. To label this protein, we incubate the sections in a solution containing a primary antibody. In this case, we are using a rabbit anti-CFOS, an antibody generated by a rabbit that recognizes CFOS. After incubating the sections in the primary antibody for a period of time, they are cycled through wash buffers and then incubated in a solution containing the secondary antibody. This antibody recognizes the primary antibody. In this case, we are using a goat anti-rabbit antibody. This antibody will bind only to the primary antibody, which you'll recall was made in a rabbit. Importantly, this secondary antibody has a fluorophore attached to it. It is this fluorophore that we can visualize with a fluorescence microscope. After completing the immunohistochemistry steps, we can mount the tissue sections on slides. Once cover slipped, they can be positioned on the microscope stage and a light source can be directed through a filter that allows only the excitation wavelength of light to pass through the tissue. This light will excite the fluorophore, causing it to emit a different wavelength of light. By filtering out all other wavelengths of light, we can use the microscope to see any parts of our tissue sample that contain our protein of interest. In this example, all of the cells that appear green are CFOS positive cells. They contain the protein CFOS. This protein was bound by the rabbit anti-CFOS primary antibody, which in turn was bound by the goat anti-rabbit secondary antibody, tagged with a specific fluorophore that could be excited and detected using our fluorescence microscope. This technique can be used to see what cell populations express particular characteristics, whether CFOS expression, the presence of enzymes involved in neurotransmitter synthesis such as tyrosine hydroxylase, or any number of other markers. In fact, with a few modifications, a single tissue sample can be labeled with multiple different antibodies, allowing researchers to see the distribution of multiple protein targets and their protein colocalization. Here you can see CFOS staining in green and beta-tubulin in red. Immunohistochemistry and fluorescence microscopy are incredibly powerful and widely used tools that have advanced the field of neuroscience.